Okay. Okay, I will read the question for you and you choose the answer, okay? Let's move quickly. Everybody in, right? I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, 11, we go. Okay, going to first question. In 25% alcohol, the alcohol occupies 25% of this. Number 1, weight. 2, value. 3, molecular number. 4, mole number. 25% of alcohol. 25% of what is included here? Okay, 6 of you gave a weight, 4 of you chose volume, 1 of you missed the time. Okay, if this is the scientific the unit, usually the percent means weight percent. Okay, if you hear 2% of something, usually that is a weight percent. It's a scientific kind of from the arrangement. If you use a special other unit, sometimes molecule number, then you say mole percent. Okay? If it's a volume ratio, volume percent. So default is a weight. Okay? So that's the question. You had some little issue for last time about soju molar concentration issues. I thought it's straightforward, but actually it was not. It's a little tricky, the point there. Okay, and move to the second question. To make 25% soju, the alcohol and water should be mixed by ratio of number 1, weight, value, molecular number, density. If you are a soju maker, if you make 25% of the soju, how do you mix? First question was, scientifically, usually there's a weight percent, but in soju company, to make it simple, they use a volume. Okay? They use a volume of 1 portion of 1 liter of alcohol, except the 3 liter of water, and to make a 25% of the soju. That's real situation. So this case is volume percent. And third question, molar concentration is a mole number in number 1 liter of solvent. Number 2, 1 kilogram of solvent. 3, 1 liter of solution, or 1 kilogram of solution. Molar concentration definition. Okay, quite good. 2 of you chose 1 liter of solvent. No, it's a 1 liter of solution, right? 1 liter of solution means solvent, solute mixed together. That's a molar concentration, the definition. Based on this, still I get only 10 answers. The density of ethanol is 0.8. What is the density of 25% of soju? Number 1, 0.8. Number 2, 0.85. Number 3, 0.9. Number 4, 0.95. Okay, it's a very democratic, so all distributed everywhere. Let's think. Alcohol, ethanol, 0.8. Water, 1.0. 25% is a 1/4, right? Then approximately you can assume 1 to 3. So which is a higher amount? Water is a higher amount. So that ratio may be around here. That's 3 to 1. So this one is a 0.95. Density could be 0.95 in ideal case. And actually in SOJU, yes, it is about 0.95. Now, next question. Same question for density of 40% of whiskey. Number 1, 0.8. Number 2, 0.88. Number 3, 0.92. Number 4, 0.95. Let me read it one more time. 0.8, 0.88, 0.92, 0.95. 40% whiskey. What would be the density? Okay, many of you get quite a good mathematically correct answer as 0.92. There is an assumption. If the volume is not changing, then this is a correct answer. The real answer is 0.95. What does that mean? In 25% SOJU case, luckily, the end result is if you mix 1 liter and 3 liter, and the result is the same 4 liter. But whiskey case, density was changed from calculated number. Calculated number was 0.92, but reality is 0.95. That means the volume is decreased or increased? It's decreased, right? Volume was originally 1 liter, 3 liter, not 1, 4 liter, 6 liter. In principle, it should be 10 liter, but it's less than 10 liter. Means ethanol molecule, water molecule, they attract each other stronger than in ethanol, maybe through hydrogen bonding, and then they made it a little smaller volume. Then many of you gave the correct answer, assuming that you calculated, Jenny calculated that 40% of weight, and then calculated those more number. Majority of you chose the volume, 40% of the 25% of volume, and then based on that, you calculate the more number, and then calculate it. You were very lucky in that case, because the volume was not changing. Now, new questions. How about the molar concentration of 40% of whiskey? So you put that answer into that same board. Another comment, Jenny gave one different answer. All of the others put almost the same answer in a row. What's the point? That panel is for open discussion. If your answer is not same from the previous your friend, what's the point to put the same answer there? That is for the discussion. If you have a different idea, or if you find something problem from others, other students, other friends, other answer, or you have a different approach, then you put your answer there. Okay? It's just for like a homework. Just I have been here and then put the same other number, same answer. It's boring. Let's enjoy. Okay? 그거 그냥 도배하는 거잖아. 그거 왜 해? When you have the unique idea, then put it up. Or you come as the first person and then put your answer there. Okay? So let's try there. Usually the problem I brought for the discussion of the panel is there is something to think. So that's why sometimes the single answer may not be just only answer, or maybe there is some hope here and there. Okay? So this is some case. I thought that this is straightforward, but when I checked those density issues, it's not so straightforward. There are some issues. Okay, next. Okay, move on. Number 60, which is not the element in DNA? Nitrogen, oxygen, phosphorus, sulfur. Which is not the element in DNA? Okay, so sulfur. Sulfur is not in DNA. DNA has phosphorus in there. Next question, which is not the element in protein? Same order. Nitrogen, oxygen, phosphorus, sulfur. Answer is phosphorus. Sulfur is in protein. This is the experiment about the Hirsch and Chain experiment, Chase experiment, right? To determine which one is the genetic material. Which is the information in gene? For lipid, for protein, for DNA, for carbohydrate. Answer is genetic information is the information for protein, isn't it? You have many of the answer for DNA. DNA itself is a genetic information, but that information is for something. 질문에 문제가 있다고 인정하지. How many genes do we have in human body? Number one, 10 million, 10,000, 25,000, 100,000, 1 million. How many genes do we have? 둘밖에 답을 안 했어? 이야, 100%긴 한데 only two of you answered. 100% correct answer. 25,000 genes in our body. Surprisingly small numbers of genes. Yeah. What is the information system in DNA? Binary, ternary, quaternary, quinary. 야, 공부 너무 열심히 안 하는 거 아니야, 이거? 너무 안 돼? 문제가 어려운가? 그래서 quaternary, ATCG. Computer system is a binary, 0 and 1. Binary, ternary, quaternary, it's a quaternary system. What is the catalyst for RNA synthesis? DNA template the DNA polymerase, DNA template the RNA polymerase, RNA template the DNA polymerase, RNA template the RNA polymerase. Okay, there is a DNA templated RNA. Oh shit, RNA polymerase. What cannot be the component in PCR? RNA polymerase, RNA primer, template DNA, building blocks with a triphosphate. Okay, the answer is RNA polymerase. That should be DNA polymerase. The trick here was primer can be RNA or DNA. Usually the primer is a DNA, but RNA can be used as a primer too, because it can make a double helix too. Telomerase template actually is RNA. Utilizing RNA, they make the telomere. What is the critical aspect of NGS compared to single sequence? Number one, cloning. Number two, speed. Number three, chemical sequencing. Number four, biological synthesis involved. Okay, many of you chose actually the HTS, high throughput screening kind of thing, but speed is a relative value. That is not the critical point. Cloning is the important concept in NGS. Let me explain this after next problem. Which is not subject of cloning? 1. Gene. 2. Cell. 3. Individual. 4. Species. Which is not the target of cloning? Gene, cell, individual, species. Okay, the answer is a species. Okay, finished. Oh, Jimmy was number one. 아, 힘들어, 씨. Okay, cloning means making exactly same copy in terms of genetic information. So cloning can be applied to one gene. Isolate out the one gene and make a copy of it and amplify. That's usually we call molecular cloning. Okay, but we also hear that if I have identical twin, that's my clone, genetically identical. That concept can be applied to a cell too. One cell divided into the other two cells, they should be genetically identical. They are cloned too. Species. You and me is the same species, Homo sapiens, but my genome and your genome may be 2% different. Then we are not clone. Clone means genetically identical body. It can be cell level or gene level or individual level. That's what we call clone and cloning. Okay? Then now it's okay about the 25% soju problem. It was interesting to think out what's the problem here. And I found there was some little more complicated situations. Okay, now today we talk about these molecules of life. Last class I tried to deliver those materials to you, and I realized you didn't have a chance to talk any. So let's start today. Any question? This week I gave you a lot of study material, maybe a little too much. That's why I didn't give you a separate assignment. I hope that you study a little more and think more about those contents. Molecules, DNA, and RNAs, and how RNA was made from DNA, and how the protein was made from the RNA, kind of a big story of the genetic flows, genetic materials flow. Any question? 내가 가져왔을 수도 있고 안 가져왔을 수도 있는데 이 클래스에서는 영어로 얘기하자. 못 듣는 사람이 있으니까.
You mentioned about native group and minor group in DNA.
Okay, that will be covered later. Okay. Other question?
And I don't think I really understand about gene and genome, the difference of them.
Okay, gene and genome. Let's move that too. So molecules of in life forms through those of the molecule in life form, so-called organic molecules. First organic molecules, at least using photosynthesis, glucose is the first product. And then utilize those glucose, we make protein. To make a protein, we need to import different elements. Glucose has only CHO. To make the protein, what other element do you need? Nitrogen you need definitely. Amino acid, every amino acid should have a nitrogen. And sulfur, we just heard. Sulfur is a part of the sum of those amino acids too. So after that, and we make, utilizing this glucose, we make the lipid and nucleic acids. So let me ask you again. Now this three is kind of, we say, three main components of the biological molecule. Nucleic acid, sometimes we count as 4, but sometimes we don't count. Nucleic acid, what is the component of nucleic acid? What's in there, nucleic acid? Is that based on protein? Carbohydrate? Carbohydrate is 1 unit, 5 carbon carbohydrate. Glucose is a 6 carbon carbohydrate. Ribose is a 5 carbon carbohydrate, but its molecular formula is the same. Carbon, H2O, and 6 or 5. Okay, it's a carbohydrate. And what else in DNA? Phosphate. Phosphoric acid, the conjugated base is a phosphate. Okay, phosphate is a linkage between the one unit and the other unit. And base or alkaloid. Nitrogen containing compound, that's a base, is a pyrimidine and purine. that we have 4 different kind of the base. That base is actually the information unit. The others are just a structural unit. Right? Ribose is all same. DNA has a deoxyribose, RNA has ribose. We will see those difference. And connection is a phosphate, and base is the information unit, ATCG. It has 4 different units. Now this linkage is a phosphate linkage, right? In protein, what is the linkage? What is the linkage part of a protein? Amino acid and amino acid. What's that? The linking part. Peptide bond, or in other terms, or you sometimes you call amide bond, CONH group. Usually amide bond is considered as a very stable bond in organic chemistry. That's more stable than ester, more stable than acid, more stable than anhydride. It's considered to be one of those most stable ones. That's how our body is composed of proteins. Then DNA versus protein. One is phosphate linkage, one is amide linkage. Which one do you think you need to keep more stably for a long time? DNA is an information system, protein is a machinery system, which should be stored for longer time. Information, right? Information should go to our offsprings, next generations. That bond should be more stable. Then do you think phosphate is more stable than amide? Chemically speaking, it sounds, is that really so? But actually it is. Phosphate bond is maybe million times more stable than amide bond. That's why Jurassic Park is possible to extract the DNA from the mosquito blood of dinosaur. It's possible, but it's not possible to extract the protein of the time. DNA is much more stable than protein. RNA is much less stable than protein and DNA. So this is basic molecules and genetic material. This question, genetic material, means we provide something to our offsprings with only 1 or 2 cells. At the moment, maybe we didn't know the identity of the sperm or egg cells, but still, we know that very small material carries all the information about us to the next generation. And that process we call genetics. And through that genetics, which material carry that information? That was a big question about 1 century ago. And the candidate was, we see those 3 major components of a biological material: DNAs, lipids, carbohydrates, and what's missing? Carbohydrates, lipids, proteins. And the candidate was, most promising candidate was at the moment the protein. Protein, we have many different kinds of protein we have seen, and it may have a more diverse something. DNA looks quite boring at the moment, and that experiment is the Hershey and Chase experiment. Very smart idea, utilizing different component, element, sulfur versus the phosphorus, and utilizing radioisotope, unstable isotope of sulfur and phosphorus, and which is transferring that information of a virus into bacteria. That's the key experiment. Of course, they got Nobel Prize, as you can guess. And now, our lipoform is a dual system. Then, 참 많은 얘기한다. Okay, our body is a dual system, and information system in DNA, and machinery system in proteins and DNA. So usually if you think something scientifically very solid, then if you do extraction, clearly separate one part DNA, one part protein, would be something more clear. But DNA is actually quite sticky material like this. If you take out the DNA from the cell, and it looks like it's a sticky material, looks like a very boring. If you analyze those components, the component is quite boring too. Protein is much more interesting and diverse. Then if you have that kind of a property of a protein and DNA, maybe protein should have more information in there, and that should carry out our genetic information to the offspring. split. That's kind of a natural guessing at the moment. So when the DNA was analyzed before Watson Crick come to the picture, this person, Erwin Shagaf, and he analyzed the relative amount of ATCG, at least at the moment that they knew. DNA is composed of 4 bases. And then whenever they compare different samples of DNA, A and T has the same proportion, and T and C and G is the same proportion. It's a changing, but at least in the same sample, almost the same content here, molar ratio, right? And same component was repeatedly discovered. So it gives you some clues. Maybe they are paired. A and T, C and G is paired. So he put this kind of information into the paper, and then later, and that DNA was somehow processed and then got this kind of data. What kind of data is this?
X-ray.
X-ray. Crystallography. DNA was crystallized, and then what technique generated this? In English, diffraction, X-ray diffraction. You make a single crystal of DNA, and then you give light in terms of the short wavelengths of X-ray light, and then that light interact with this crystal, it generates diffraction pattern. And this is the first diffraction pattern data made by Rosalind Franklin. And then by using that data, we can retro transfer that information into three-dimensional position of molecules. So that is the result of double helical structure. At the moment, it's not very sophisticated model like this. It just shows that looks like it's a helical. Utilizing this information here, it has these people, and they analyzed it, whether this is a 2 helix or 3 helix. Double helix or triple helix was the key issue at the moment. And this was one team in England. And the other team is the Linus Pauling in US. Linus Pauling was already Nobel laureate at the moment. He discovered the structure of a protein, alpha helix structure. And he should be genius person, and he has some data like this too, but he predicted that structure as a triple helix. And these guys, they found this one is a double helix. And during the weekend, they wrote this one-page paper in Nature, and they got Nobel Prize for this. They typed in, at the moment, there was no computer. It's middle of the 20th century. And they typed in and drew this picture and put it in Scotch tape. I don't know whether they had a Scotch tape or not. And then they sent this into Nature. And then for this, they got Nobel Prize. Okay, this is the DNA structure. Now you heard about the minor groove or major groove. Groove means the small shallow dorang. I cannot think the other word. So that groove is actually depending on those the positions. This is major groove. The groove is not localized only one position. One side is a major groove, and the other side is a minor groove. Double helix is not symmetrically going in, and then one line, the other line goes a little shifted one way, and then one side is major, the other side is minor, and major groove is moving down like this. Same way, minor groove is also moving. This is minor groove. Okay. That is the groove situation. Groove is a long line of this DNA. Everywhere there is the grooves. Okay. Open side and then a little shallow side. So those grooves is provide that anchoring position. If you add some small molecule, they can bind selectively here. Major groove is more difficult because it's an open structure, so it has a big the open space. It's difficult to bind. Minor groove is shallow, so some molecules can easily bind to minor groove. Okay. Okay, general question. Major groove, minor groove, I just explained. So that is along line all those DNA there is. Information in the gene, that is gene and protein. Basically, gene information, the quaternary information in gene is the information to make protein. Okay, DNA is in a sense it's a linear information, ATCG, and protein is also in a sense that's kind of linear, the first product. Okay, but protein is composed of 20 amino acids, 20 units. DNA has only 4 units. Now, how much information can be stored in DNA? That makes the decision of how many genes should define one amino acid. So to calculate that, let's think about the binary system, computer system, 1 and 2, and each one unit we call bit. Right? This is a binary bit. Now we hear this is a qubit, quantum bit, but that's a different story. So if this is a binary bit, if there is a 2-bit, 2-bit means the 2 unit. 2-bit, then 2-bit can carry how many informations? 4 information. 1, 1, 0, 0, 1, 0, 0. It's a permutation, not combination. 조합이 아니고 순서가 있으니까. So 0, 1 and 1, 0 is different. So that's a 4 information. If it's 4-bit, then how many information can you store? 16. Then with the 4-bit, can you define 20 amino acids? Still not yet, right? You need more. 8-bit, how many information can you get? 16 by 16, what's that?
256.
256, okay? 256. After that, 512. 10-bit, 1024. At least up to there, we have some the idea that we hear that number quite often, right, in computer. So 2 to the 10, to calculate this simply, 2 to the 10 is 1024. This is similar to 10 to the 3, right? So if you calculate it quickly, let's say 2 to the 30. To get the idea, you can break this 2 to the 10 to 3. Then this is about 1,000. Thousands 3 times. So that's million, billion. Okay, this is the way you can convert 2 to the something into ternary system. Okay, approximately. Now, if you look at this, then if you have n bit, the information number is 2 to the n. You can think. Then to code 20 amino acid, what is the smallest number of the bit?
5.
If it's a 5, then you can cover 32. Then that's enough numbers to cover all 20 amino acids. But you need to think that if you have this kind of a binary system, then you need to know which one is the starting point. Right? So start and end point you need to put separately. So for example, maybe all 0 or all 1, maybe this is the starting point and this is the end point. You need to assign at least the 2 sequences separately. Right? So comparing to this, let's come back to DNA system. Now we have a 4 unit, 0, 1, 2, 3, quaternary system, not binary system. Quaternary system, it has a direction, so that's why it's not the combination, it's a permutation. So from the 5 end to 3, the 3' end, this is direction usually we read. And here, if it's a one bit, 1 nucleic acid, that's this is a 4 information. It's a 2-bit, 4 by 4, 16, and 3. Now that's 64. Then to cover 20 amino acids, how many is the minimum DNA? 3, right? In a sense, a little too much. Maybe a lot of waste in there, but still, there you can take out the one starting code, start codon, and end codon, stop codon. Still, there are 62 you can use. So those 62 is distributed to different amino acids with a different ratio. And simple amino acids has more copies. And complicated amino acids, like essential amino acid, usually we hear, 필수 aminosan, something we need to take, expensive amino acids, then usually it's a smaller portions. Makes sense, right? So at least 2 code 20 different amino acids, we need to have at least 3 bit. And then this code can be matched to amino acid sequence. And this is a genetic code, And then this is matching to amino acid sequence here. 3 is matching to 1 amino acid. That makes sense, right? So direction-wise, amino acid, there's amino group and acid group. If you make a protein, there's one end to the other end, and then the direction is genetic information-wise from 5' to 3'. Protein case from N terminal to C terminal. That's the direction usually we draw this way. Okay. This information is stored in DNA, is made into protein. So if you describe our biological computer system, now you hear time to time 32-bit computer or 64-bit computer, right? So that means 32-bit is designated to assign one information. But sometimes the information number is too big, then usually we use 64-bit. That's our current computer system. 32-bit or the 64-bit computer is designating how big of one unit of information we utilize. Our system is a quaternary system, but 3-bit computer systems. 3-bit quaternary system. Your computer, 32-bit binary system. Okay, that's information system-wise we can describe our genetic information. So for this, let's say if you need to make protein, which has 100 amino acids, then how long of your DNA sequence required? Minimum 300. This is matching to this, right? 300 sequence of DNA can designate 100 amino acids. Actually, you need a little more. If you need to do PCR, you need to have a primer sequence. Right? It should be extra sequence here and the end positions. So a little more sequences you need. And anyway, this one is so-called. If this nucleic acid is making exactly protein, then this area we call coding DNA. Here, code means coding protein information. That's the coding region, coding DNA. Okay. When you studied about the genome, the project in our body, genome is a collection of genes. One gene, by definition, one gene is the information to make one protein. Genome is a collection of all those genes together. So defined by, let's say, human genome. All the genes together in human DNA, that's genome. Om means it's omnibus. It's a collection of everything. Funny thing is, after we have finished the Human Genome Project, we have a 3 billion long DNA. Out of those whole DNA, this is the coding area, which is designated to protein sequence. That's only 2%. The other 98%, something, other functions they may have. Even 10-20% of them is repeated sequences, which has a very similar sequence to virus. Maybe virus infected our body, they put their gene into our genome, and then they make multi-copies in there. And virus is multiplied and in our genome. And they occupy 10% of our genome. Ridiculous, isn't it? Still, they may have some function, or maybe that's kind of just a scar. We got infected some time ago by some virus, and then their virus sequence is left in our genome. Now, those 2% of a gene is kind of a classical gene, which make a protein. And leftover 98% of those DNA, sometimes we call junk DNA, but more and more we figure out that the function of those, the undescribed ones. Mouse has much shorter, maybe 10% to 20% of the genome size of a human. That means total number of this gene number, classical gene number is similar, 25,000. Surprisingly small number. Bacteria, they have 6,000 genes. And fruit fly, chopari, they have about 20,000. C. elegans, the worm, they have also 20,000. We believe proudly our body is much more complicated, and our emotion, culture, this should be all encoded into the gene. We should have a lot more numbers of genes, we believe. maybe we should have a minimum 100,000 numbers of the gene. Actually, we are not very much different from, not to tell the monkey, it's a rat, mouse is very similar to us, and we are even closer to the chocolate, the fruit fly. 20,000 versus 25, what's the big difference? Now those uncoding area has a lot of difference, and then more and more we found the new functions of them. About half of those, 2% make it protein. The other 50%, they are transcribed into RNA. At least up to RNA level, they make it. And then after that, they do not make a protein, but they have their own function. The other half, even they do not make RNA. But if you delete them, there's some problem happens. Means they have some functions. Now, I said the classical gene here, the other 98% of this DNA's function, maybe we should call them as a gene too, because they have their own role and functions, even though they do not make a protein. Okay, DNA to protein. DNA is the information to make a protein, and then if you look at that, utilizing this DNA information, if it makes a protein, that's what we call gene expression. Gene is expressed. It means that gene information is utilized to make the protein. And this is not directly done. I said it's a dual system, but there is a DNA and there is a temporary information of RNA, and then that is moved to the protein. And first step here, transcription and translation. In a sense, that's almost the same thing. Transcribe means by looking at something and you write down, that's a transcribe. Translate is reading some foreign language and then write it to the new language. Well, makes sense. It's a good description. DNA, RNA is almost the same molecule. It's almost like writing same copy. That's a transcription. Translation, information is designated, but DNA and protein is very much different molecules. That's translation. Quite a romantic description. So that's a transcription and translation. Now this is your DNA and this is RNA. Can you catch the difference? It's almost the same molecule. And this is ribose. Ribose is exact the carbohydrate. This is ribose. Okay. 6 is the glucose. Now that is the original form. And if you look at DNA, we say DNA is a deoxyribose. Deoxy means ribose is a template, and then oxy is removed. That's a deoxy. And these small changes make a big difference. Phosphate bond is connecting this unit to this unit. I said this phosphate bond is very stable, even more stable than amide bond in peptide and protein. By the way, this molecule, RNA, is a billion times less stable than DNA. Why do you think? By looking at the structure. This OH group can attack this phosphate group and then push this out, so-called similar to SN2 reactions. It's possible. It's a neighboring effect. Very close attacking group right next to phosphate can attack and cleave this bond billion times faster. DNA does not have that group in here. So that small differences make a big difference. So DNA we use for permanent record of our genome. RNA we make the temporary information, make one gene unit each, and then use that information to make the protein. And it's chemically unstable, biologically unstable too. So this RNA molecule, after it makes protein, it is degraded, removed from the body. Makes sense? Right? So for now, we have dual system, DNA and protein. RNA is a kind of intermediate material. Now, if you look at the structure and DNA, to make its a copy, it needed to replicate. To replicate it, you need to copy something means you need to have a template. That's why they chose this kind of a template system matching to each other. It has hidden information over the other line, but gene information is not both of them. Gene information is on one side. But DNA replication, you need to make two copies. So you have seen, you have studied, it should be divided into two, open up, and then make the synthesis. Synthetic process is only from the 5' to 3' directions. One is smoothly can move on. The other one cannot, right? That's why it has some fragmented the synthesis. And some Japanese scientists predicted that may happen. What's his name? Okazaki. And by that prediction, and he got Nobel Prize with another experimental person. Somebody else proved that, yeah, really Okazaki fragment, the system is happening. Because of that, this fragmentation system, the end position of the DNA is difficult to be fully synthesized. That's why telomere is shortened and shortened every step of the cell division, right? So using this DNA synthesis, the DNA polymerase, DNA templated DNA polymerase, right? RNA synthesis, DNA templated RNA polymerase. Then other option is possible. RNA templated RNA polymerase. Do you think it's possible? In our body, usually we don't have. Junyoung said yes. Can you give an example? RNA templated RNA synthesis happens where? RNA virus. RNA virus, they use RNA as their genome. And then they need to make their genomic copy. Coronavirus, HIV virus, those are RNA virus. We just said RNA is super unstable compared to DNA. There are DNA viruses too. DNA virus is closer to us, utilizing DNA as genetic information, and they make RNA to make a protein. But some viruses, they use RNA as their genome. Much more unstable, somehow they protect their genome, and then they survive, and they evolve. And that's why RNA virus usually it's more unstable, and usually their mutation speed is faster than DNA virus. They utilize this instability for fast evolution. So next week you are going to study about evolution process. So that will be something you may think. So here, DNA polymerase, RNA polymerase. One more thing. RNA templated DNA synthesis is possible. Any example? Reverse transcriptase and reverse the transcription. Some RNA virus, they use their genome RNA, make DNA, and insert that DNA into the host genome. The host genome make a lot of copies of the RNA. So this is utilizing host the machinery system and then make their genomic copies. That's also possible. All process was discovered so far. RNA to protein, the catalyst is ribosome. That ribosome is not protein, that is RNA. That's why there was some hypothesis about RNA world is made from this observation. Maybe ancient time. Already we have seen there are RNA viruses that exist. That means RNA is unstable, but they can act as a genome. And some of those functional molecules can be also made by RNA. Then, maybe ancient time, before we divide the information and machinery system separately, maybe there was some ancient life forms. It's RNA based. RNA genome and RNA machinery. That's the hypothesis of RNA world. No evidence, but just a speculation. Now we have more RNAs here. This is also RNA. This is also RNA, and this is the information system, and this is a machinery system, right? This is catalyst, ribosomal RNA or ribosome, and then we need to give different name for this. So this is a messenger RNA, and there's another important RNA, tRNA, transfer RNA, which carries another information here. But anyway, RNA can do many different functions. This is mRNA is information. This is kind of an enzyme. Okay. Now, one more picture. DNA double helix And then when mRNA is made, it made a copy of this, and this is the mRNA. So this is, let's just say this is one gene. Open up partially, DNA is very long. Let's say this is one gene, then open up some partial area, so-called the transcription factor, determine the starting point, and open up, and then make this mRNA here. Some of you asked the question that if a double helical, double strand, the RNA has a problem, then how do you control it? In the body, they make only one side, this RNA, they do not make this side. So usually, naturally, we don't have double helical RNA in our body. Only viruses they have. That's why if you recognize the double helical RNA, we feel that we got infected by virus. Then we do something extra function to remove that. That is the basis of siRNA and microRNA. Okay. So using this, let's just say, if you see this 5' to 3' direction synthesis happening, now this is sequence here. If this is sequence, if you read the ATCG sequence here, if that is a matching to genetic code, matching to amino acid, we call that sense strand. Opposite side here, this one into here, this is a template for this. This is what we call anti-sense strand. For example, sense strand is AAAAA. The anti-sense should be TTTTTT. AAA and TTT carries a totally different the designation for amino acid. So template anti-sense sequence, information-wise, it doesn't have anything with the protein sequence. That means only one strand is meaningful, and the other strand is just a template for synthesis. Then, which strand, let's just say this is the upper and lower strand. Which strand is used for mRNA template, upper one or lower? Actually, it's mixed. Sometimes this area going this way, here, here, another area going this way, this way, this way. Those genes are back and forth. It's a mixed, but they do not make both of them together. Partially overlapping is possible, but they do not make the same copy to make the different genes. Okay, mRNA carries a sense. Then this template here, template should be anti-sense, right? So mRNA is the sense because that makes the sequence of matching to the protein. So this template should be anti-sense, or you can call it as complementary strand. So that's why you have heard the term of cDNA. cDNA means if you get mRNA collection and then make its complementary copy of DNA, then that sequence is a DNA by using the reverse transcriptase. From mRNA, you make DNA, then that's complementary DNA, antisense DNA. But there is a template to make more mRNAs, correct? What do you mean? If you wanted to collect the information to make a protein from one cell, you collect all mRNAs. Those mRNA, and then you make reverse transcriptase, you make a cDNA. Then those are the template to make more mRNA, right? So mRNA is unstable. You make a cDNA and collect all together. All collection we call it library. cDNA library is from one cell, the whole mRNA synthesizing information collection. That's a cDNA library. That sequence is a template sequence, complementary antisense sequence. Okay. Okay, I think it's almost at the end. Intron, exon, then how the live cells they can recognize which part is the intron, which part is the exon. Well, this intron, many of them, it has its own folding and the ribosome-like functional role they have. It's like a ribosome, ribozyme. And then they form its structure, and then they cut, and then connect those exons. So intron is not just a sequence. Many of those introns, they have functional roles. Or sometimes there's other helpers, like transcription factors. They can have two, but many of those introns, it's not only sequence. It is kind of a functional materials. Okay, here the telomerase case. So because of this, the Okazaki the fragment, telomere can be shortened and shortened. How about we add telomerase to make it longer and longer, and we can live forever. That's one good idea. We can live forever. Our cells do not get aged, but that increases the chance of getting more cancers. So very well controlled. Living long versus aging is kind of balance. So cells cannot divide forever, and approximately 50 times they can divide by experimental the observation. And telomerase is one of the important factors, but just boosting those telomerase is a little dangerous because it can boost the cancer production. Okay, I think this is the last slide. So again, we see next week. Next week the topic is evolution. So you learned about the genetic information here, DNA, RNA, and then how to the evolution happens, and then how do you utilize the evolution into the experimental setup in the lab, so-called molecular evolution. That will be the topic of next week. Any question for this week?
Sure. The rivals, the hydroxy group protects the phosphate SN2 reaction. I thought that some groups will fall apart too. And could you give additional explanation of the SN2 reaction after the SN2 reaction?
Actually, it's not, technically speaking, it's not SN2 reaction. Just this is a kind of a nucleophile which has a high electron here, high electron, low electron. So high electron, the oxygen can attack this position, and it can be compete. This is oxygen, this is oxygen. So whether this is connected or this is connected, that's kind of competition. So this oxygen can attack here and then push this out. Then the end result is that this linkage is broken and making this cyclic phosphate here. So anyway, the key point is reactive group is nearby. That reactive group can destroy those strands. That's the key point. Other question? Yeah.
You said in the meetings that RNA to RNA bonds are stronger than RNA to DNA than DNA to DNA. So is that caused by the hydroxyl group?
Well, if you ask, okay, let's review them first. So if they have the complementary sequence through Watson click the base pairing, through hydrogen bonding, A and T they can form this kind of the attraction. CG they have 3 bonds, so CG has a stronger the attractions. Now if you look at those position of hydroxyl is here, right? So actually DNA, RNA, it doesn't matter. The major interaction happens through base to base. And then this is a little far from this base pairing. So in principle, DNA is most common double heavy cover interaction. DNA RNA also happens, and by chance we observe the DNA RNA is even stronger than DNA. RNA is even stronger. That's observation. As a result, we can speculate that maybe this hydroxyl somehow that affects maybe this angle or the conformation of this site. It's favorably contribute. But that was designed or not, that's a different story. Because we don't have many DNA, RNA, or RNA, RNA decoupling in the cell. It's just an observation. Any other question? Okay.
While you're having a lecture, I'm certain that how to distinguish the major groups and minor groups. How should I?
If you look at here, actually the position wise, so this strand is a 2-dimensionally put, right? Actually, this is a twisted. And then one side is have the opening space this side, the other side is opening here. So this is the minor group and this is the major group. So because of this structure is not identical in both sides. Twisting is not equivalently, symmetrically going on. This is going one side like this. So that's why one direction is giving a little bigger than the other because of that asymmetry. So this is a big one is flowing down here, right? And then this is a small one going down. If you look at from this structure, this is one side this way, this is the other side of this way. So depending on the ribose positions, which side is major, minor is defined by ribose structures. Okay. 말로 설명하기 어렵고 모델을 보면서 그렇게 잡들다고 생각해 봐야 돼. Even for me, it took several months to fully understand. I cannot claim that I can fully understand. When I look at those structures, it was quite difficult to understand. But by looking at that, anyway, the major groove is flowing down all the way. Exactly the opposite side of the minor groove is this is major. So those are the facing to each other like this. Okay, another question. Okay, it's better. You guys give a little more questions. I will try to give you more chances to give questions. And then today, let's finish here.